We developed an innovative method for metabolomics. We combine label-free spectral imaging of living cells with single-cell mass spectrometry. Our technique can monitor and predict the metabolic changes of single cells against drugs. And this opens a lot of application for basic research and industry.
Our method adds single-cell resolution to current drug evaluation techniques, which will greatly aid future drug discovery efforts. Our technique can also help us understand the role played by cellular heterogeneity in cancer resistance.
The single-cell sampling and the data analysis are the most challenging aspects in this experiment. We are currently working on automating these two steps, especially the sampling part. We recommend you practice the single-cell sampling well before the experiment, as each micromanipulator setup is slightly different, and you should take the time to get familiar with the controls.
After incubating cells to reach 70% confluency, culture cells of interest in an appropriate culture media. Add penicillin streptomycin to avoid contamination. After measuring cell density on a hemocytometer, subculture cells into a 35 mm glass bottom grid dish or quartz slides using the same medium at a seeding density of 0.7 × 10⁶. Then, incubate at 37 degrees Celsius for 24 hours. The next day, the cells reach a confluency of 50 to 60%. Wash the cells 2 times with pre-warmed PBS buffer at 37 degrees Celsius